All right, ladies and gentlemen, so welcome back. So today we're going to pick up, we've been talking about these action potentials and sending these signals down the axons, but what actually happens to that signal once it reaches the end of the axon? And that's where these neural synapses come in. So when you look at this end of your axon, you've got these junctions, these synapses that happen from one nerve cell either to another neuron, so the information can be passed neuron to neuron, or it's some other effector cell, so neuron to muscle or neuron to gland, for example. When we look at these synapses, you really have two options. One can be an electrical synapse, where we take that action potential down the axon, get to the synapse, and then cause an additional electrical flow from one neuron to another. This typically is going to involve the flow of certain ions from that synapse to the next neuron. But overwhelmingly, most synapses are chemical. And so we get down to the end of that axon, and what we do is end up causing the release of certain neurotransmitters. Those transmitters then are information that diffuses across that junction from one neuron to the next cell. So here's what we're looking at. When we look at chemical signals or chemical synapses, here's your axon of that presynaptic cell. This is the one that action potential has just moved down, and we're now at that synaptic terminal. Here is your postsynaptic cell. This could be another neuron, this could be a muscle cell, this could be a gland, but it's some sort of effector cell. It's your postsynaptic cell. So as that action potential moves down that axon, what happens when we get down here is that your cells have produced these synaptic vesicles containing neurotransmitters. That action potential will cause those neurotransmitters to get released. Those neurotransmitters then diffuse across that synaptic cleft, which is what this little junction is here, this little gap is here. And on the opposite side, on that postsynaptic cell, are ion channels, receptors, something that's going to receive that neurotransmitter and then cause a change in that postsynaptic cell itself. So after the release of that neurotransmitter, it diffuses out into the synaptic cleft. When it does so, it can be taken up by that surrounding cell, either on a receptor like we saw right here on the cell membrane that can cause change. It can also potentially diffuse straight through the cell's membrane and have a receptor internal to that postsynaptic cell, or they can also be degraded by enzymes. And realistically, all three of these are going to happen. It's a question of what happens when, how quickly does each particular process occur. So when we look at those postsynaptic cells, really the question is, what is the change that I'm going to cause in that postsynaptic cell? If this is some other direct synapse that's causing those transmission of those neurotransmitters to bind directly to ligand-gated ion channels like we see back here. All right, so this would be a ligand-gated ion channel, right? It's moving the sodium and potassium. There's a receptor here for a ligand. In this case, it's specifically the neurotransmitter that's working here. Then those neurotransmitters cause ion channels to open and then have the potential to set up another potential change in that postsynaptic cell, specifically if we're talking about neuron to neuron information. So those postsynaptic potentials really then end up falling into two categories. Am I going to cause that next postsynaptic cell to also have the potential to send another action potential, in which case we would talk about an excitatory postsynaptic potential? I'm going to cause that next neuron to also depolarize, potentially getting that cell to a point where I'm moving it away from its resting potential. And if I hit the threshold of depolarization, I can also cause that postsynaptic neuron to also send another action potential down its axon and kind of repeat the process. This one's going to involve both your sodium, potassium, and calcium ions. So those are the three ions that get involved if I'm going to have an excitatory signal being sent from one neuron to the next. You can also have a situation where rather than it exciting the neuron, trying to send another action potential, I can actually hyperpolarize the neuron and pull it further away from the potential for sending a signal. That's where we get into hyperpolarization, and that's where we involve things like your calcium and potassium ions. So I'm causing the resting potential to increase. I'm getting it less likely to send a signal as opposed to more likely to send one. So this table is just kind of nice, gives you an opportunity to sort of compare and contrast action potentials within those postsynaptic cells. So it goes over a little bit of the excitatory versus inhibitory and what it does, what's its function, what does it mean in terms of feedback loops, all sorts of things. So nice one to kind of take a few minutes and take a look over. I encourage you to do so. What we need to understand, though, is that when we look at your postsynaptic cell, more often than not, it's not just getting a single signal from a single other presynaptic neuron. Instead, it's more likely getting signals from multiple neurons at once. And so the question then becomes, am I talking about all excitatory, all inhibitory, or is it some summation of both? So if I look at a situation like this, if I've got two postsynaptic neurons that are both sending in excitatory signals, well, the question then becomes, what is the summation of that information? So in this case, I've got only that neuron 1 sending in excitatory signals. One signal kind of dies off, then it sends another one. So I never sum together, they never come together enough to actually get me past that threshold, and so an action potential never gets sent. On the other hand, if I have that same excitatory impulse coming in, even if it's coming in from a single neuron, but they're coming really close together, then I may get to a point where they sum up and send the signal. So it's sort of like waves. So having one wave come in and die off, one wave come in and die off, it's never enough to actually sort of knock you over in the ocean. But if I start sending waves really close together, the signal may become strong enough to actually send that action potential. And in this case, take that wave analogy. Now the wave crashes, knocks you over, and that's sort of where that action potential comes in. In that same mind, I can also have excitatory neurons. The more that I send in and the faster the signals go, the more likely I get to that threshold and send that action potential. But I can also have a combination of excitatory and inhibitory signals coming in. So if I've got an excitatory and then an inhibitory, it's like they cancel each other out. And so I need to overarchingly have enough excitatory information coming in to overshadow that inhibitory if they're both working at the same time. So in order to send that signal, regardless of that combination of excitatory and inhibitory, they need to be able to make sure that I send that signal. So it's ultimately the sum of everything coming in that determines whether or not that action potential threshold gets met. So in some synapses, right, we can have a neurotransmitter that binds to that receptor that is metatropic, meaning that the movement of those ions depends on one or more metabolic steps. So the binding of that neurotransmitter may activate a signal transduction pathway inside the cell that involves some sort of second messenger that may also cause some other changes to occur. But compared to those ligand channels, these second messenger systems are a lot slower. But on the flip side to that, they last longer. So it's a little bit more like an endocrine signal in a neuron as opposed to just a strict electrical one. So if I look at metatropics where I involve that second messenger, slower onset, but last longer in terms of the signal as opposed to just being the movement of ions and opening up those gated channels instead. That'll happen and send a quicker signal, but the effects will last not nearly as long. So it really depends on what the cell's goal is as to which process it decides to utilize.
What's 1,000 times thinner than a piece of paper, more numerous in you than grains of sand on a beach, and proof that the smallest things can sometimes be the most powerful? I'm talking about the synapse, the meeting point between two neurons. If your neurons form the structure of your nervous system, then your synapses, the tiny communication links between them, are what turn that structure into an actual system. Because as great and powerful as your neurons are, when it comes down to it, their strength and their purpose lies in their connections. A single neuron in isolation might as well not exist if it doesn't have someone to listen to or talk to. The word synapse comes from the Greek for to clasp or join. It's basically a junction or a crossroads. When an action potential, and if you don't know what an action potential is, watch the last episode, sends an electrical message to the end of an axon, that message hits a synapse that then translates or converts it into a different type of signal and flings it over to another neuron. These connections are rather amazing feats of bioelectrical engineering, and they are also ridiculously, mind-bogglingly numerous. Consider that the human brain alone has 100 billion neurons, and each of those has 1,000 to 10,000 synapses. So you got somewhere between 100 to 1,000 trillion synapses in your brain. Each one of these hundreds of trillions of synapses is like a tiny computer all of its own, not only capable of running loads of different programs simultaneously, but also able to change and adapt in response to neuron firing patterns and either strengthen or weaken over time depending on how much they're used. Synapses are what allow you to learn and remember. They're also the root of many psychiatric disorders, and they're basically why illicit drugs and addictions to them exist. Pretty much everything in your experience, from euphoria to hunger to desire to fuzziness to confusion to boredom, is communicated by way of these signals sent by your body's own electrochemical messaging system. Hopefully, you know enough about email and texting etiquette to know that if you're going to communicate effectively, you have to respect the sanctity of the group list. It's not a great idea to send a mass text to all of your friends first thing in the morning to give them the urgent news that you just ate a really delicious maple bacon donut. Seriously, people, if you happen to have a friend who truly adores bacon, then an email would suffice. But if you're out clubbing and suddenly Bill Murray shows up and starts doing karaoke, then that would be a totally appropriate time to notify all of your friends at once that something awesome is happening, and they better be a part of it. And in much the same way, kind of the same way, your nerve cells have two main settings for communicating with each other, depending on how fast the news needs to travel. Some of your synapses are electrical. That would be like an immediate group text. Others are chemical synapses. They take more time to be received and read, but they're used more often and are much easier to control, sending signals to only certain recipients. Fortunately, your nervous system has better text etiquette than your mom and knows when each kind is appropriate to use and how to do it. Your super fast electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. They're super fast because the signal is never converted from its pure electrical state to any other kind of signal the way it is in a chemical synapse. Instead, one cell and one synapse can trigger thousands of other cells that can all act in synchrony. Something similar happens in the muscle cells of your heart, where speed and team effort between cells is crucial. This seems like a good system, so why aren't all of our synapses electrical? It's largely a matter of control. With such a direct connection between cells, an action potential in one neuron will generate an action potential in the other cells across the synapse. That's great in places like your heart, because you definitely don't want half a heartbeat. But if every synapse in your body activated all of the neurons around it, your nervous system would basically always be in group text mode, with every muscle fiber and bit of organ tissue always being stimulated and then replying all to the whole group, which would stimulate them even more until everyone just got maxed out and exhausted and turned off their phones for good, which would be death. So that would be bad, which is partly why we have chemical synapses. They are much more abundant, but also slower, and they're more precise and selective in what messages they send where. Rather than raw electricity, these synapses use neurotransmitters, or chemical signals that diffuse across a synaptic gap to deliver their message. The main advantage chemical synapses have over electrical ones is that they can effectively convert the signal in steps, from electrical to chemical back to electrical, which allows for different ways to control that impulse. At the synapse, that signal can be modified, amplified, inhibited, or split, either immediately or over longer periods of time. This setup has two principal parts. The cell that's sending the signal is the presynaptic neuron, and it transmits through a knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5 millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channels to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited. The neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message onto the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area. Now we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around and keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or more alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters. And sometimes they can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets 3 major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize moods by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate, and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober state, you got all these neurotransmitters doing their thing in your body, but once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because you have a limited supply of these neurotransmitters, and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all of those synapses remember how great those extra chemicals feel. As a result, you actually start to lose receptors, so it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong, and when it comes to your synapses, that, my friends, is what wrong looks like. In their natural healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, a science. Today you learned how electrical synapses use ion currents over gap junctions to transmit neurological signals, and how chemical synapses turn electrical signals into chemical ones using neurotransmitters before converting them back to electrical signals again. And you learned how cocaine is a sterling example of how artificial imbalances in this electrochemical system can lead to dysfunctions of all kinds. This episode of Crash Course was brought to you by Logan Sanders from Branson, Missouri, and Dr. Linnea Boyev, whose YouTube channel you can check out in the description below. Thank you to Logan and Dr. Boyev for supporting Crash Course and free education. Thank you to everyone who's watching, but especially to our Subbable subscribers like Logan and Dr. Boyev who make Crash Course possible. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale. The script was edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe.